is a holiday. So then exam over 13, 14, and 15 is on Wednesday. So we're in the immune system here. Remember that the immune system is divided into two parts, the non-specific side and the specific side. Another name for non-specific is innate, and another name for specific side is acquired. And then within each of those, we've got three lines of defense. First and second are part of the non-specific side, and third line of defense is within the specific side. So, remembering, first line of defense, what is that?
Skin.
Your physical barriers, that which keeps the pathogens out. And there's a whole bunch of them, right? Skin, tears, mucous membranes, mucus itself, cilia, blah, 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 blah. If these get broken, then we move on to the second line of defense. And what's involved in the second line of defense?
Inflammatory.
Inflammatory response, in which our damaged cells produce cytokines. They release cytokines. Those get secreted from the cell. They make blood vessels leaky, extra leaky. That extra leakiness helps the macrophages and the neutrophils squeeze out of the capillary, enter the tissue spaces, and then eat up whatever is there non-specifically, right? There's also complement here. Whoops. Complement is for bacteria only. But as far as the inflammatory response specifically, in the second line of defense, which cells run phagocytes? Those that do the eating. Okay? Then we've got our third line of defense. And the third line of defense has two sides. We've got the antibody mediated. Who runs that? B cells, because we're making antibodies. And then we've got our cell mediated side, and that's controlled by T cells. All right, so kind of ended up at this point yesterday, I think. So remember that our B cells make antibodies. B cells have to recognize that there is an antigen, that there is something foreign that has invaded the body. And B cells can find this, can identify this pathogen in one of two ways. One, they can bind the antigen directly, meaning B cell has antibodies standing on the surface, IgD type antibodies, and they can bind the antigen directly and recognize it and say, okay, now this B cell says, all right, there's the antigen, I need to make a whole bunch of these antibodies so that they can fly around the bloodstream looking for these antigens and target them for destruction. So they can bind directly. Second way that a B cell recognizes the antigen is to be presented antigen by an APC, an antigen presenting cell. So remember that an antigen presenting cell has phagocytized the antigen, chopped it up into little pieces, and then stuck a piece of that out on the cell surface via that MHC protein. Then that antigen presenting cell can show it to the B cell, and then the B cell says, all right, now I see this antigen, I need to make a whole bunch of antibodies that will recognize that, send them out into the bloodstream so they can target it for destruction. Remember, are antibodies little grenades that blow things up? No. Do they kill? No. They frost, they surround. Targeting for destruction by macrophages. It puts that extra yummy frosting on the antigen so that your macrophages And neutrophils, your phagocytes, can find it and eat it faster and get rid of it. It also covers that antigen with the frosting, the antibody frosting, so that this antigen can't stick to your cells. Viruses have to bind to your cell to enter. So if we can block the binding, you block the infection. Even bacteria, before they can start dividing in your body, have to bind to your tissues before they can start to divide. So if we can block the binding, we can block the infection. All right. So once the B cell recognizes the antigen, what happens?
Get stuck.
He's stuck. Can't do anything. Whoop-de-doo. That's no fun. So there we are. We're stuck. We are what's called sensitized. That's a fancy word. Sensitized. We're stuck. T cell side. We've got different types of T cells. One, which is of the T cell family, although really isn't part of this third line of defense, is called the NK cell. We said the NK cell really lives over here in the second line of defense because it's one of the patrollers. The NK cell is like the scout, right? Running around looking for the bad guys coming in, waiting for them. They bind to, no, they don't bind to, they recognize cells that are infected with virus or cells that are cancerous. Two different things. So here's the NK cell. Here's the cell that's got the virus in it with a little flag out there. It says, I'm infected, I'm infected. And the NK cell sees it and then spews out these poisons that result in apoptosis of the infected cell, but also we've got some collateral damage as well, right? But they're out there all the time. They're out there working. They're of a T cell family. We talk about them here, but they really do their work over here. So these are our shotgun shooters. Collateral damage. Second cell is a CTL, or a killer T. They also recognize virally infected cells, but instead of having a bunch of collateral damage here, we've got precise killing. So here's our killer T cell, and it binds to, it literally hooks up to the infected cell, and then spews out chemicals directly to the one cell, killing it one by one by one. These guys are slow. They're slow to respond. Why? Because they have to be told to go by somebody. And who is that somebody? The general, which is the helper T. So the helper T cell is the one who's going to tell the killer T, or the CTL, go out and look for virally infected cells and kill them, please. Helper T cell is the general. He recognizes that there is a pathogen in the body because who told him so?
The APC.
Antigen presenting cells who phagocytize the one thing, chop it up, and stick a piece of it out on the cell surface, and then they leave their tissue spaces and they go into the lymph where the T cells live, and they show it to the different T cells, looking for somebody that's going to recognize it and get activated and jump on the bandwagon here. Remember, any phagocytic cell can be an antigen presenting cell. A neutrophil can be an antigen presenting cell, a macrophage can be an antigen presenting cell, but that's not their only job, right? Their main job is to eat stuff. Dendritic cells, that's their only job. The only job in their world is antigen presentation. So we recognize the antigen, And then what do?
They tell Dorothy to go find
How?
by cytokine.
They release cytokines. They release lots and lots and lots and lots of chemicals. And those chemicals, those cytokines, tell the CTL, get out there, get to work. The cytokines tell, whoops, well, that's true. Tell our phagocytes and our NK cells, keep up the good work. The helper T also then goes to find this stuck B cell, binds to it, and gets him unstuck. And once that B cell gets unstuck, then what can he do? What does he turn into? Turns into bunches and bunches of plasma cells who make bunches and bunches of antibodies. And those antibodies are specific to the thing that the B cell recognized. They're specific to the antigen that the B cell recognized. Now, do you think that only one B cell is working here in the process? No. Let's say we've got a bacterium that's got these 3 different antigens on the surface. There can be a B cell that recognizes this one. There can be a B cell that recognizes that one. And there can be a B cell that recognizes that one. So now what you're going to make is all these B cells are going to get activated in the process. So each one is going to make its own antibody. One's going to look like this. One's going to look like this. And one's going to look like this, right? So it's a multi-pronged attack. It's not just a single antibody. It's a single antibody to the one organism, right? It's the one thing. It's the one bacterium. It's the one virus. But you might make a little bit alternating antibodies that recognize the surface antigens of that organism.
Does it take a while for the body to make the antibiotics?
Once they do this, it takes a while because they get stuck. But once they turn into plasma cells, it's speedy. Yeah. And the amount of antibodies they produce is something like each one makes 2,000 antibodies per second.
So when
That's speedy.
when it's stuck, the helper T cell tells it to become plasma.
Tells it to turn into a plasma cell. Right. So, let's look at the overall picture. I didn't think we got to the picture, did we? I know, it's hard because they're combined. The classes are combining, I know. We're doing the same thing in micro, so. The image that I have for this one, which is the interaction between the antibody-mediated and the cell-mediated side, remember, these work hand in hand. This is the third line of defense. The third line of defense has 2 parts to it. Can you do one without the other? No. Can you have a B cell side and make antibodies without the T cell side? Can you do the T cell side and have an effective response without the B cell side? No. If you didn't have any B cells, you'd have your killers out there shooting. Whoop-de-doo, right? You got a bunch of poisonous chemicals floating around, killing pathogens. But you wouldn't have the antibodies to block infection, and you wouldn't have the antibodies to frost and make them yummy for your phagocytes. So they have to work hand in hand. They go hand in hand, and this is sort of the overview of it. But let's look at it.
Is there anything that causes one side not to work than the other? Hold on.
Dang. This image came from the internet. I didn't like the one that was in your book, so I found this online and I altered it a little tiny bit. So this is the B cell side. So we've got these flip-flopped here. Here's the B cell side. There's a B cell, and it's going to recognize the antigen directly. So the antigen is the little yellow ball there. Recognizes the antigen directly, or it can be presented antigen, right? I added that in. Once it recognizes the antigen, he is sensitized, which means he's stuck. Okay, so there he sits. T cell side, macrophage, antigen presenting, or APC, could be a neutrophil. Chews up the antigen, sends it to the lysosome, chops it up, and instead of spitting it out this time, we're going to stick it out on the cell surface. And then what we're going to do is we're going to run around and we're going to look for, we're going to leave the tissue spaces, and we're going to enter the lymph, and we're going to look for a helper T cell. Now, this is a crappy picture just because it looks like a B cell, because don't those look like antibodies? Okay, but it's not. So pretend, just pretend that these aren't there. So we'll just go like this. We'll just give them a little fuzzy outside, right? So there's our T cell. Gets presented the antigen. And now that's our helper T cell. Now he's activated, and as soon as he gets activated, he starts spewing out cytokines. Those cytokines activate your CTLs, your killer Ts, further activate your macrophages, further activate your NKs, tell them to keep up, do the good work, tell the CTLs, go out, find out what's going on, and start shooting things directly. Then what does he do? Then what he does is he goes over, this is still our helper T. I'll get rid of these little antibody looking things. This is still our helper T. He's going to bind to that stuck B cell. Literally, goes and looks for it and binds to it. Hooks up to it, releases cytokines so that he can get unstuck, turn into tons and tons and tons of plasma cells who release tons and tons and tons of antibodies. Our helper T also turns into a memory T cell to remember that we've seen this before. And the B cell also turns into a memory B, so we can, if we see this again, we can fight it quicker. The importance of these memory Ts and memory Bs is that if they're here and they're working and they're staying alive for length of time, sometimes they last your lifetime, sometimes they don't. If they're there functioning, then the next time you get infected with that pathogen, these guys recognize it, and we don't have to wait for presentation. We won't get stuck. If the B cell recognizes that antigen from the pathogen, immediately turns into plasma cells making antibodies. If the memory T cell recognizes that antigen, immediately spews out cytokines, tells your CTOs and your macrophage and your NKs, go, keep it up. And the response is more aggressive and much faster than the original response. This takes time, right? It takes time to eat the antigen, find the T cell, present it to him. T cell has to find the B cell, tell him, okay, go ahead, get unstuck. This takes time. It takes effort. You get sick before you get better, right? You get an infection, you go through the illness, that's the point where we're doing all this stuff. We're building the reaction, we're building the immune reaction. And then once this is all activated, that's when your illness starts to wane because your immune system is now winning the battle.
So where does HIV?
Well, just where I was gonna go next. You're reading my mind. What happens if you don't have that cell?
We can't really cytokine to unstuck the B cells.
You can't do this. So you can't do this. So you can't do this. And you can't do this. And you can't do this. So what do you have?
You have B cells that can't get unstuck.
Well, what do you have for an immune system? You got your second line of defense. You got your macrophages and your NKs out there, or your neutrophils, your phagocytes, and your NKs. That's it. That's all you got. Which is why people with HIV, untreated HIV, ultimately die. Because they get infections that kill them. Infections that if your immune system were healthy, wouldn't be a problem. They're considered massively immunocompromised. After the point where their T cell gets to be dropped below what's considered 200. So your T cells should be in the range of about 1,000 per mil maybe, I don't remember the numbers. But if your T cells drop below 200, you don't have enough to do the work.
So with like the medications like we see Descovy being advertised a lot lately, does it just help fight the HIV or does it help
Yes.
keep your...
No, it blocks HIV replication and
So it blocks it from going into the cell
and making more of it to go out. And because what HIV does is it enters your T cells, your helper Ts, gets replicated, release HIV to go get more helper Ts, but it's typically a slow process. It takes up to 10 years for
So
T cells.
HIV basically just targets the T cells?
T cells mostly, and some macrophages too. So then if you got low macrophages, you really have like 0. And they're like I said, massively immunocompromised. So they succumb to, infections that somebody else could normally fight. They've got lots of very good drugs out there now for HIV that dampen the amount of virus. In fact, at some point, some people who have been on some of these drugs, research has shown that the virus is completely undetectable. Normally, the virus is pretty low in the bloodstream because it's hiding in your T cells. But they've now suggested that potentially these drugs could cure.
I think there's very, very few people that's been, I think I just read saying that one girl is now became
HIV so free,
Free.
right? Cured. So that's the hope, that you could actually take these drugs, completely knocks out the virus to the point that it's no longer there and detectable, and she should be able. Could she go off the medications and be considered HIV free? I don't know.
So if someone,
That's the question.
if it's super low that's undetectable, can they still give someone the virus?
That is the level of medications that they have now, as I understand it, is that if you are HIV basically undetectable, then your partner, you shouldn't have to have, protected sex, and your partner sometimes is on prophylactic HIV medications to prevent infection, and they're saying that maybe they can just go to normal. So, 2 different pictures, you guys in micro, you pick your favorite picture, doesn't matter to me. Like best. You guys got a lot of immune system these last couple weeks here.
What does leukemia?
Leukemia is a cancer of the white blood cells,
White blood cells.
depending on the type of leukemia. So, we said that immune memory produces the involves your T's, you're building your memory cells, your memory T's and your memory B's. Primary response is the first exposure. We're making the memory T's and memory B's. Second time you get infected, second time you get exposed, it doesn't necessarily block the fact, it doesn't necessarily block infection. It's just that the response is so much more aggressive and so fast. We don't have to wait for presentation. We don't have to get stuck. Your memory cells immediately respond. And so here's your primary response. This is just antibody production. First primary response, you make antibodies. You continue to make antibodies over the couple of weeks. Peaks out about here, and then your antibody production wanes. You fight the infection, and the level of antibodies always drops. These are considered the neutralizing antibodies. This is the frosting that you're making that's going to block the virus from attaching to the cells. It's going to block the bacteria from attaching to the tissues. It blocks the infection. It always wanes. So in COVID, if you ever remember hearing about the neutralizing antibodies, what's your antibody level? What's your antibody level? This is the antibody level they're talking about. And then the concern was, well, the antibody levels have dropped after infection, right? After like natural infection or even vaccination. Well, the antibody levels have dropped, so that means we're not immune anymore. Not necessarily. They always drop. So if you listen to some of the doctors and scientists, it's supposed to drop. They're supposed to drop down to lower levels. That doesn't mean that you don't have memory T's or memory B's that are sitting there just waiting for COVID to come again and spew out antibodies. If you have good natural immunity or immunity, if you have good memory cells, especially memory B's, look at how fast this memory B cell starts to respond. Here's infection day. Within the first day, the memory B cell says, oh, look, I've seen this before. Watch this. Divides into bunches and bunches of plasma cells who make millions and billions of antibodies immediately. Neutralizing antibodies. These are the frosting. So now the virus doesn't get a chance to infect the cells. It doesn't blow them up. It doesn't cause infection. You don't get sick. You don't even know you've been infected. And that's the purpose of immunity. Again, those wane over time.
What causes them to not be in your system a long time? Is it just the body's way of saying, oh, we don't need them anymore?
It's the type, it's the disease. It's the disease itself. Number one, if it's a virus that mutates and changes its surface structures, then it looks totally new. It's like a whole new virus. That's what they're concerned with COVID, right? The new variants, is the immune system going to be able to recognize it? Well, I don't know. We're going to have to find out. It's also kind of the level of infection. So the worse you have the disease, the sicker you get, the better your memory cell response, the better memory cells you make. If you got a wussy COVID infection, asymptomatic, but you tested positive, sure, you probably made some memory cells, but maybe not that many, and they might not live very long. If you ended up in the hospital and you were sick, really, really sick, and were there for 2 weeks, and you get out, your immune memory is going to be better than somebody asymptomatic. At least that's the going thought. Allergies, we talked a little bit about allergies. Allergies are a hypersensitivity to normally innocuous, meaning antigens that shouldn't cause your immune system to even recognize and have a response. We're not supposed to be, pollen should not activate our immune system. But it does. And a lot of that is due to your genetics and things like that. Your first exposure to the antigen, or to the pollen, whatever, you don't have a response. So if you're allergic to whatever, to like shrimp or seafood or something, you have to be exposed to it first. And then you make your memory cells, and then it's your second or subsequent exposure that you get the allergic reaction. Remember, these are of the IgE type. So IgEs are the antibodies that stand on the surface of either mast cells or basophils. Just waiting for that antigen to land and activate, bind to the IgE, and cause the release of histamine. Anaphylaxis just means you're having an allergic reaction.
So what do we like grow out of allergies?
You can get desensitized.
Is that what?
Like you can get allergy shots, which is usually best with things like grass and pollen as opposed to peanuts and nuts and stuff. And what they do is they give you little tiny bits of the allergen, and the goal is for your body to make IgG antibodies, which then would bind up the antigen before it could bind to your IgEs and have the allergic response.
My mom's friend had that, and now the little girl can eat up to 8 peanuts, I think.
No peanut butter and jelly sandwich.
Not yet. But it's been years that she's been getting these shots every so often with the small particles.
Yeah, it's not as effective on food allergies and stuff as it is on pollen type grass. So don't worry about the differences between delayed hypersensitivity. Just means the reaction is delayed, and it's a different mechanism. It's a T cell mechanism as opposed to an antibody mechanism. Immediate hypersensitivity is you eat it or you get exposed to it and you immediately have a response. We talked about transplants, talked about HIV, autoimmunity. So autoimmune diseases are diseases usually associated with a viral infection. And so you get infected with a virus. Viruses like to have these little spikes on them like COVID. You get infected with a virus and you build antibodies, right? Because that's what we do. We build antibodies specific to the proteins, covering it, frosting it, blocking it from infecting, making it yummy for your phagocytes, right? So now you've got all these antibodies floating around that are specific to the virus, but in some cases, these antibodies have cross reactions to your body tissues, like classic one is the myelin sheath. So for whatever reason, these antibodies see proteins on the myelin sheath as foreign, but it's after a viral infection. It's usually after a viral infection. You make the antibodies, which you get rid of the virus, and then X number of amount of time later, these antibodies bind to your myelin sheath and target it for destruction. And then you have a disease called MS. I read an article the other day and it's on my desk somewhere. Article the other day about new research that indicates that MS is associated with herpes infection. So it's an autoimmune reaction to herpes. 85% of the population is infected with herpes and doesn't even know it. So it's a very common virus. Why do some people get it? You know, why do some people have that cross-reactivity and some people don't? Again, it's probably a susceptibility based on genetics. Pick your parents better. Always say. One of the goals of cancer is to, and we're going to watch a video on cancer in lab So I'm not gonna go too much into this. If you wanna watch this video when you get home, you can. I'm not gonna show it now, but one way that we're working on targeting cancer, besides just giving nasty chemotherapy drugs, is to get the immune system to recognize the cancer cells better and kill the cancer. The immune system doing it, yourself. But it tends to be very specific, meaning each person, everybody's cancer is different. So then you'd have to, develop a technique individually for each person's cancer, which is extremely expensive, and FDA doesn't run like that. FDA runs like, we're gonna have this drug that treats everybody equally, not individually. Because if you do it individually, each individual drug concoction has to go through clinical trials, which take years, and yet ain't nobody can do that. But if we can use some immune system cells to target generalized structures that are common on many cancers, that would help. Chemotherapy drugs. If you have cancer and you're gonna go get chemotherapy, what chemotherapy does is targets fast-growing cells, because that's what cancer is. Cancer grows too fast. It's fast-growing, fast-multiplying, messed up, whacked out cells. And
Is that
so,
why your hair falls out?
yes, because not only does it target the fast-growing cancer cells, it targets the fast-growing body cells, which include your hair, and that's not hair in your head, it's every hair on your body. You're bald. It affects your GI tract, because the lining of your GI tract is replaced quickly.
Is that why they vomit a lot?
They get GI upset, they get terrible heartburn, diarrhea, red blood cells, white blood cells, they drop. So now they're immunocompromised. Yeah, it's basically you're poisoning the person. You're poisoning the cancer, but you're poisoning the person at the same time. So we can use the immune system. And there are some immune drugs out there. And if it ends in, if it ends in MAB, N-M-A-B, there's a bunch of drugs out there. Immunolobulab. They're just crazy words, right? If it ends in MAB, that's an immune drug. That's a drug where we're activating the immune system. And they're out there, and some of them work very, very well. But they're good for very specific cancers. Okay. Now remember, I'm gonna start 16, but this is not gonna be on your test next Wednesday.